Okay, in this video we're going to talk about neural integration. Integration refers to neurons functioning together in groups, and these groups can contribute to kind of broader neural functions. So of the billions of neurons we find in our central nervous system, a lot of these need to integrate information in ways that allow for a nice, smooth, and coordinated response. So when we refer to neurons that function together in groups, those are going to be pools. So pools of neurons can receive and integrate incoming information from other neuronal pools, and they basically process and forward this information along to other destinations. So a simple neuronal pool is basically we have a single presynaptic fiber that branches and discharges on several different neurons within their pool. Now the discharge zone refers to neurons that are closer to the incoming fiber and are therefore more likely to generate an impulse. The facilitated zone refers to neurons that are more on the periphery of the pool, and because they're farther away from the incoming fiber, they're usually not excited to threshold unless you have other neurons that are stimulating those cells as well. So to see this represented pictorially, we see that we have an axon coming down here, and there's lots of axon collaterals here that are branching, and those have their teleodendria here. And essentially what we find is for those neurons that are in the center and kind of closest to the axon here, these ones are in the discharge zone. They're more likely to fire action potentials because they get more inputs. If you count up all the inputs here, you see that those neurons in the center here have a lot more synapses than those in the periphery, and because of their many inputs, they're more likely to fire and generate their own action potentials as well. For the neurons that are off to the side, because of their fewer inputs and they're farther away from the rest of the fibers, these are much less likely to fire action potentials unless they're actually stimulated by other cells as well. Now, other types of neuronal processing include serial processing. This is actually where we have input that travels along one pathway to a specific destination. Now, one neuron can stimulate the next one, which stimulates the next one. And the system works in an all or none manner to produce very specific and anticipated responses. And a best example of this is like a spinal reflex, where if you tap the patellar tendon, there's an impulse that goes back to your spinal cord, then that activates a motor neuron that then stimulates the appropriate skeletal muscle to contract, which then causes your leg to kick out. Now, reflexes are rapid, automatic responses to stimuli, and they occur in an all or none process. Once you stimulate a reflex, it should complete. Now, a particular stimulus always causes the same response, so that's why we say that these reflexes are stereotyped, because they produce the same response every time. Now, they occur over pathways, which we call reflex arcs, which have these 5 essential components. We got receptors, sensory neurons, the integration center, motor neuron, and the effector. So to kind of look at what a simple reflex arc would look like, we'd have the receptor level, which would be somewhere in the periphery. That's going to generate action potentials that then are traveled along this unipolar neuron back towards the spinal cord, where integration occurs, and this is where you get communication with an interneuron. The interneurons are multipolar neurons that receive many different inputs, but in this case here, it's just a very simple input. And this interneuron then can synapse with a motor neuron, and this motor neuron can transmit action potentials out towards an effector to carry out the response. So in the case of the patellar tendon reflex, we see that if you tap the patellar tendon, this activates stretch receptors in that tendon, which then can generate action potentials that are transmitted towards the spinal cord. You get a communication between the sensory neuron and your interneuron to motor neuron, and then you get action potentials out towards your muscle, which then causes your leg to kick out. So with parallel processing, we see that input travels along several different pathways, and different parts of the circuitry deal simultaneously with the information. So this is a little bit more complicated than serial processing, but with parallel processing, this is important for kind of higher level mental functioning. And an example of this could be something like a smell that reminds you of an odor that could be associated with other experiences. Maybe there's a smell that reminded you of a fond memory from childhood, and smelling that causes the memory to become conscious again. Now, these circuits here can occur in a lot of different ways, but there's 4 major types. We have diverging, converging, reverberating, and parallel after discharge. And so what we see here is a diverging circuit. Diverging circuits are essentially where one neuron could branch out and activate other neurons that then activate other ones, which activate other ones. So you see that the signal then diverges. So from one neuron, you can get many different neurons that are activated. And think of this as like amplifying information. So an example of diverging circuit where information can be amplified would be like in the spinal cord. We could have one motor neuron that connects to many hundreds of muscle fibers or even thousands of muscle fibers. And so that even though you have one neuron that's activated, it can go to lots of different outputs. So a converging circuit is where you have many inputs, but they all converge on one output neuron. And you can see here you have like 3 different neurons that are then synapsing on one output neuron here. And in that sense, it's converging. And you can think about this as basically an information consolidation, where an example of where this occurs in the body is like different sensory stimuli can elicit the same memory. You know, for instance, these neurons could be from like the odors of, you know, freshly cut grass, and this neuron might be activated when you smell maybe like wet cement, you know, and this neuron might be when you see, you know, maybe like a bright sunny day. And because of these converging information, you know, you get this output that then could potentially allow you to remember, you know, maybe just a summer evening that you had in the past. So a reverberating circuit is essentially where a signal travels along this chain of neurons, and there's actually feedback along the way. And so what's interesting here is that these reverberating circuits create rhythms or oscillations. So you might wonder, well, where in the body would you want to have neural networks that generate rhythms? Well, it turns out that we actually have a lot of rhythms in our body. You know, there's a sleep-wake rhythm. We have breathing rhythms. There's also repetitive motor activities like walking. We also have neurons in our brain that help keep time, and they do this by generating a certain rhythm of action potentials, and this gives us a sense of time, like how much time has passed. And so these reverberations create these frequencies and oscillating circuits that essentially allow us for you know, rhythm generation. And the parallel after discharge circuit is where a signal stimulates neurons that are arranged in parallel arrays, and they eventually converge on a single output. This is basically where an impulse reaches an output cell at different times and then causes a burst of impulses called an after discharge. So if you look here, we would find then that because this one motor neuron has an axon that then directly synapses on the output neuron, this is going to be the fastest. And then over here, where you actually have 2 neurons in the path on the side here and the side here, these are going to be slower. So you're going to find then that although the action potential is going to get to this neuron quicker, other action potentials from these pathways will also get to the output neuron. And this creates kind of frequencies of action potentials that you know, eventually stimulate an output neuron. So with these parallel after discharge circuits, you can get more kind of odd patterns of output. An example of this would be things like exacting mental processes like mathematical calculations. You know, like understanding mathematical equations could rely on these sorts of parallel after discharge circuits. So in terms of the developmental aspects of neurons, we know that our nervous system originates from something called the neural tube. And the neural tube actually comes from the layer of ectoderm embryologically. And this tube is basically where the ectoderm folds and essentially makes the CNS. So your central nervous system is the folding of this ectoderm from the neural tube, and the neural epithelial cells of this neural tube proliferate into cells that are needed for development. Neuroblasts become amitotic and migrate, and these neuroblasts can sprout axons and connect with targets to become neurons. And this occurs pretty early on in development. Now, the way that neurons can form connections is by something called a growth cone. And the growth cones are kind of a prickly structure at the tip of the axon that allows it to interact with its environment through like cell surface adhesion proteins, neurotropins that attract or repel the growth cone. We have nerve growth factors or NGFs that can attract these growth cones, which help keep these cells alive. And then filopodia, which are growth cone processes that follow the signal towards a target. So think of the growth cone as like this sort of extending process that is guided by lots of different chemical factors until it reaches its target and it can form a new communication point with a target cell. So once the axon finds its target, it must find the right place to form a synapse. Now what's cool here is that astrocytes that help guide the development and growth of neurons, they actually provide the physical support and the cholesterol that's needed for the construction of the synapse. So the astrocytes play as a nice sort of intermediary role with helping to facilitate the communication between different cells. because they essentially give the appropriate components for that synapse to form. So about 2/3 of neurons actually die before birth, which is fascinating. So you actually have a lot more neurons in your body before birth than after. And if these axons don't form with their target, then they're triggered to undergo apoptosis, which is programmed cell death. Now, many other cells also undergo apoptosis during development. So what's interesting is that later on after birth, what you're left with and what you develop with is actually a far fewer number of neurons than what you were born with, or even what you had prenatally. Now, these neurons are amitotic before birth. However, there are a few special neuronal populations that can divide after birth, especially in areas like your olfactory neurons, in your olfactory bulbs for smell, and your hippocampus, which is involved with learning and memory. These are some of the few areas that where you still have nerve cell division, and you can generate new ones to replenish maybe aging or damaged neurons. So in terms of what the growth cone looks like, you can see that there's the axon, it has its expanded tip, and all these kind of projections that hang off here. Now on these projections here in the growth cone, you actually would find receptors for different chemicals, and the chemicals that these receptors pick up either guide or inhibit the growth of this cone in that direction. So you can think about these little projections as being like a little nose that sniffs out the correct chemical scent trail for where this axon needs to connect. And I think that's pretty amazing.